All right, hey biology. Hope you're doing well when you watch this. So this is lecture 3 in our, DNA unit, and this is all about the central dogma of biology, like the central thesis, the central thought. And so DNA is transcribed in RNA and translated into protein. This is how flesh and blood, like proteins and different phenotypes, come from a code A, T, C's, and G's. How does that happen? Well, how powerful is DNA? It's crazy powerful. You have something called Hox genes, and these are developmental genes. Not going to really get into that. It's more like a upper level course, but basically these genes help the body develop into what it needs to be. What they did is they took a gene that helps develop in the legs and they put it in the head, like the Hox gene of an antennae or the head of the fruit fly. And you can see heads growing out of its body. They did this in a lab. DNA is that powerful, right? And so our objectives in this part is to contrast RNA and DNA and then to explain the process of transcription. And so it is important to see like the phenotypes come from genes, but genes are made up of DNA. And so these two tigers have different fur color because different genes, right? And this tiger has a low fitness level because it won't be able to blend in very well in the environment. Okay, so DNA is the boss. That's from A Wonderful Life. That's Mr. Potter, the kind of the antagonist in It's a Wonderful Life, a Christmas movie. And so DNA is the boss, but how does it get the message out? DNA is like if you have blueprints to make the Batmobile, and there's the Batmobile. Like, how do you actually make it? What do you actually do? And so DNA, yeah, this is a kind of a common analogy in science. Like, it's the blueprints, right? And so DNA has to make these RNA molecules that go to ribosomes, which make proteins. And so RNA is kind of like a working copy of a gene, okay? And they control so many different things. So you have to be able to tell me the difference between RNA and DNA. You have to tell me the different sugars, ribose versus deoxyribose. You have to tell me the different bases, uracil versus thymine. Apple in the tree, car in the garage for DNA. Apple maybe in the umbrella or under, I'm not sure. But A binds to U in RNA. Double-stranded, DNA is double-stranded. RNA is single-stranded, like a one-way street. And then, yes, there's a double helix in DNA. So an analogy that I've been using in class is like a pizza. I love homemade pizza. It's hard for me to eat like, you know, Domino's or Papa John's after you make like homemade pizza from scratch. It's just nothing like it. So let's say I have an Italian cookbook, but and this glorious recipe passed down for generations. But I don't read Italian, so I need to change that into English to make my pizza, right? And some of you might be like, well, yeah, you're going to use Google Translate and translate that, but I want you to use the word transcribe. Scribes write. And so we're going to write it in a different language, Italian into English, and then we're going to assemble the ingredients, and then we're going to assemble, once we have our English, recipe, we're going to assemble the ingredients in the kitchen and build and bake our pizza. And it's going to be delicious. And so that's kind of the analogy, right? So much like DNA is in a different language than RNA because different bases, and then RNA is used to build the protein, and that's the flesh and blood we talk about. And proteins are enzymes and structural components and all sorts of different things. That's an analogy to think about. We're going from Italian into English into pizza. And so messenger RNA, they carry information, usually encoding for a protein. Now, remember, only 1.5% of your DNA is encodes for proteins. So there's like 98.5% doing something else that you'll learn about down the road in your education. But carry information from DNA to other parts of the cell. So this is the recipe, okay? The ribosomal RNA is kind of like our kitchen. This is where we're building the protein. And so proteins are built, rRNA, right? rRNAs mix up ribosomes, and ribosomes are also made up of proteins, and they have an enzyme function as well. But ribosomes build proteins, okay? And then transfer RNA brings amino acids to the party. Okay, amino acids are it comes from your diet, what you eat. Okay, there's roughly 20 essential amino acids, and they all are brought by different tRNA molecules. And so the amino acid structure, like the amino acids are put together, these are the monomers of a protein. So different amino acids would make a different protein or a different shaped protein. So I gave you this in your notes. just highlight a few things here. First off, highlight gene. All RNA comes from genes, all of them. rRNA, mRNA, tRNA, they all come from genes. Some people sometimes forget that, like, oh, just because we're talking about a gene, we're talking about a protein gene. No, no, no. There's genes that offer all sorts of stuff. And so rRNA makes the ribosome, mRNA builds a protein for our purposes, and tRNA brings all these amino acids. Preferably, these would be different colors because they have like different, shapes. They have different R groups. You'll learn about that down the road. But basically, they have different shapes. And depending upon which amino acids are put together, this protein takes on a 3D shape. And certain shapes work well, certain shapes work a little bit, and certain shapes won't work at all. And this leads to phenotypic differences, what we see in life. And so all of these arrows right here are transcription. And then this right here is translation when we're building our protein. Okay, so mRNA goes through the ribosome. Let me get the pointer here. So mRNA goes through the ribosome. Ribosome reads every 3 letters. We'll talk about that in a second. tRNAs come in and bring amino acids to the party. And then the ribosome puts those amino acids together. And then however many amino acids it takes, you have that. Okay, and so this is transcription. Notice that RNA polymerase builds RNA. It's involved with transcription. It reads A, T, C's, and G. It reads this line, okay? But it builds with a U. Notice when it reads an A, it builds a U. It can read a T, but it puts an A. So this, again, this is RNA. This pink line is RNA being made. Our transcript, our instructions to build our protein. How important is this? Here's ATP synthase. You can't live without it. ATP. You need, I don't even know, like trillions upon trillions of molecules of ATP. And so there's like 14 or so genes that make up this molecule. Every single gene needs to be transcribed and translated correctly for this molecule to work. Okay, and so there's a central dogma of biology. So transcription, RNA polymerase builds RNA. It reads the DNA and builds an RNA transcript. Okay, just to get you a little touch of maybe down the road, in prokaryotes, there's only coding regions, which means the regions we need to build the protein. But for eukaryotes, there's introns, which is kind of like a regulation, being able to regulate what kind of proteins we make. I don't want to get into this too much. And so basically, a vocabulary word is you want to remove the introns. You don't want them in. They don't need to be in the final mRNA transcript. You keep the exons. Exons good, introns removed. Not necessarily bad, but right now you could think of them like, hey, we don't want them. Okay. This is like extra information. So this is like in the recipe book, intron would be, in the analogy, like in the Italian recipe book, this would be like if the author goes into it and starts talking about like their first memory eating pizza with their grandmother. And then they continue the recipe, and then they stop and they start telling you about the best restaurant they've ever had pizza at in their hometown in Italy. Like all of that is introns. We don't need that information to build our pizza. We might need it down the road for whatever reason, but we don't need it right now. Okay, and so go ahead and try to answer these questions on your own. Go ahead and pause it, and I'll tell you the answers. So pause, answer. Okay, so introns is extra, instructions, which would lead to extra amino acids, which possibly could lead to a protein that doesn't work. Okay, I'll just give you the first 9 here. So if transcribed mRNA is this, what was the template strand of DNA? Well, it's 3 prime. Again, we're not going to worry about the primes. Sorry, I teaching all these things. Let's just do the first 3. A U G C U C G U G. Okay, so if RNA, right, this is RNA, what was the DNA that it came from? Well, it read a T to put an A, it read an A to put a U, C, tac. This is G, A, G. Notice C's and G's are always the same. This is C. If it put a U, it read an A. A binds to U, an RNA, and a C. Okay, so this is transcription. Okay, and here's RNA versus DNA. So there's 26 letters, of course, in the English language, 1 million plus words. And so much like that, there's words in the genetic code. Okay, and then I showed you the video about the gentleman who became a genetically modified human, GMOs, right? Genetically modified organisms. Well, they are now treating sickle cell by changing DNA in people. Gene therapy. Crazy to think about. And so this is, how it could be a game changer for sickle cell disease. And so why does sickle cell happen? Because we have a different code. And that code leads to differences. Okay, so the genetic code in RNA is translated into the amino acid sequences of proteins. So translation, where we take the code, happens at ribosomes. We're taking mRNA and building a protein, or we're taking the English cookbook, English recipe, and building our pizza. In the analogy. So in prokaryotes, since there's not much space, they're coupled, but of course in eukaryotes, nuclear envelope separates mRNA production and translation. So in the genetic code, there's something we read in every 3 bases, and these are called codons. There's 64 codons. There's 64 words in the genetic code. And we led to this. This is a beautiful thing. And this is like years and years of research, how they figured this out. But basically, science now knows, and they've known for a while, when the code is GUU, we're going to build with the amino acid valine. Okay. When it's ACA, we're going to build the amino acid threonine. And proline is all of these 4. Okay. So you can see that one codon leads to one amino acid. Sometimes there's numerous codons that encode for the same amino acid, but still 1-to-1. It's never like one codon can encode for multiple different amino acids. That would be ambiguous. And so let's practice this. All right. Let's practice this right now. So CCT, so let's transcribe this. So CCT would be GGA, GAA, this is AUG, and this is UGU, right? Only U's in RNA. And then what would the protein be? Well, you come up here, and the first one is GGA, right? And so you get G, G, so you kind of go across here, come down, meet where the 2 column and rows meet. GGA is right there. It's glycine. Okay, so then you would just write the first 3 letters, glycine with a dash. GAA would be glutamic acid right there. Okay, and so that's GLU. AUG is methionine, and UGU is where? UGU. There it is. Cysteine. Okay, CYS. Okay, do this on your own. We'll talk about it in class. Okay, and so a pretty crazy thing is here's the largest gene in your body if it opens up correctly. Let's see, still recording the screen. Nice, I guess it will. And so this is the largest gene in your body. This is called titin. It helps your muscles have elasticity, which means they return to their normal shape after you've contracted them. Look how big this is. These are all A, T, C's, and G's. This is crazy. This is how complex you are. And this has to be transcribed and translated. Again, crazy. The largest protein in the human body, or at least that's what I remember from my education. Titin. Crazy stuff. Okay, and so how many amino acids does each codon represent? How many codons can code for a single amino acid? Well, this is multiple. Multiple different codons can code for the same amino acid, but this is 1-to-1. One amino acid is represented by one codon, okay? And so translation, yeah, I'll show you some videos in class, but basically, translation happens. There's initiation, elongation, termination. I'm not probably going to quiz you on that. What I will test you and quiz you on is mRNA brings the instructions to make the protein, rRNA builds the protein, and then tRNA is bringing amino acids. And then how the amino acids structure, these are all amino acids. The amino acid structure determines the completed polypeptide. Shape determines function. Okay, and this just kind of summarizes what we've talked about. So most genes contain instructions for assembling proteins. Many proteins are enzymes which catalyze and regulate chemical reactions. A gene that encodes for an enzyme to produce a pigment can control the color of a flower. Proteins are microscopic tools designed to build and operate. Okay? And so this is the central dogma, DNA to RNA to protein. Of course, many exceptions. For example, retroviruses or HIV, their genetic code is actually RNA and stuff like that, but we're not going to really dive into that right now. And so it's universal. When you say that word, especially in like the secular biology, like that points to, they want you to think evolution. In a Christian school like we are in, we can think, common designer and, stuff like that. We'll talk about that in the evolution unit. And then gene expression means like what, genes can be turned on or off. Like a muscle cell versus a nerve cell look completely different, but yet they have the same DNA. So how does that happen? Well, certain genes are being read and transcribed and their products being made and certain genes are not. Here's eyes on legs. Here's Hox genes that are problematic and stuff like that. So these are just all examples of gene expression, which for our context, we don't need to go into right now. So this is central dogma, right? One more time. DNA is transcribed into RNA and translated into protein. Hope this was helpful. We'll talk more about it in class. Thanks for watching.